Shall we begin? It's 5:02. Thank you for joining us this evening. I'm Michelle Griffin, and I'm your host for our call tonight. I am a volunteer with JDRF. I am on the Greater Bay, formerly known as Greater Bay, now Northern California board member. I was also on the International Board of Directors for 6 years. So I want to thank you for all you've done as supporters of JDRF. We have some of our greatest supporters on the call tonight. And when I think about where we're at, it's incredible how the science and technology and the people have all come together to put us in this exciting place that wouldn't have been possible if not for all of you on this call and all of those families and individuals who went before us. So from a mother's heart, I am grateful to you. My volunteering began when my son Cameron was diagnosed back in 2007. He had just turned 9. He's now 22. And he's given me permission to share with you that as a young adult, living with T1D has been really hard for him. And as his mother, of course, I want to do everything I can to change that. When he was first diagnosed, I remember that year, we didn't know anything about T1D. I remember that year thinking, cure, cure, cure. Like that's all I could think about, right? We've cured this 250 times in mice. Why can't we cure it in people? And I came to learn over time how complicated, how sophisticated a disease T1D is. So I put cure more to the back of my mind and really focused on living better with the disease for the next 10, 12 years. And then 2 years ago, my dear friend, Karen Jordan, started to talk to me about the center. And I could not have been more excited. And Aaron Kowalski started to talk about cures plural, which is not language that I had heard before. And I am so thrilled that my family has been one of the early investors, we like to call our donations to JDRF, one of the early investors in the center. And I hope when we're done with this tonight, that you too will feel as excited about the center as our family does. I'm about to introduce my dear friend, Karen Jordan, who's going to moderate this session for us. A couple of housekeeping before that. We're on obviously Zoom format. We can't see the participants other than the panelists, and participants are muted, but please use the chat function to submit any questions that you might have, which will be handled in the Q&A period. So back to Karen. So Karen is she was past president of our Northern California board, is a member of the International Board of Directors, and is the chair of the research committee, among many, many other things that she's done. And she's really a shining example for me of how much impact a volunteer can have at JDRF. She's simply amazing. And so I am going to turn it over to Karen at this point. Karen.
Okay, I always forget to unmute, so I'm going to start again. Thank you so much for the introduction, Michelle. I always say that I'm following in your footsteps. So I appreciate the example that you set for me and for all the other volunteers. Oh, sorry, I'm getting a message. Okay, got a message from the host. I am excited to share the Center of Excellence initiative with all of you here, and I appreciate you making time to join us this evening. It's a relatively new initiative for JDRF. You probably saw today, we announced our third Center of Excellence in New England. So we now have 3 centers, but the Northern California Center is the first center under this initiative. And really the goal is to accelerate progress toward the cures, as Michelle was saying, with focused funding supporting unique expertise at different institutions. You know, the other great thing I think about this initiative is that we will enable collaboration among all these different initiatives. And if you saw the announcement about the New England Center, you'll see that there's overlap and very complementary activities happening at both centers. And in fact, there are a lot of ties among the scientists at UCSF and Stanford and the scientists at Harvard and the other New England institutions. What makes Northern California unique, and this center in particular unique, is that it's bringing together 2 world-class institutions, UCSF and Stanford, and really setting up the platform to enable the close collaboration and to bring these complementary areas of expertise together to work on finding cures for type 1 diabetes. So the primary goals of the center are 3, and they are really built on the proven principle that we know one way to cure type 1 diabetes, and that is through islet transplantation. There are people today who have received these transplants and have remained insulin-free for years. So we know this approach works. And this is something that UCSF and Stanford are building on, and they're doing it in 3 ways. The first is understanding how the immune cells and the islets and beta cells interact. The second is developing islets and immune cells from stem cells. And the third is building off a transplantation protocol that's been developed at Stanford, where they're able to transplant organs without requiring long-term immunosuppression. And then just taking a step back, this center structure is something that is appealing because it takes away that whole administrative burden on the scientists. They don't have to reapply for grants and go through the whole approval process. They now have, with this center, guaranteed funding for 5 years, and they have the flexibility to be able to change direction, to build on findings without having to go through a very formal reapplication process. And it really allows them to spend the time in the lab working and dedicating their expertise toward the progress that we all are hoping for. So with that as kind of the framing, I'd just like to quickly introduce our 2 scientists so that we can really get to the stars of the show. And I'm going to start with Dr. Meyer. Everett Meyer is using his expertise in immunology to help develop cures. He is a recipient of JDRF's Career Development Award, and he's working on a method to modify the immune system to protect islets and teach the immune system to tolerate them. He has a very personal connection to type 1. His son was diagnosed just a few years ago. And he is working with Dr. Judy Shizuru, who's also part of the center, who is actually using elements of this protocol in human clinical trials. Dr. Julie Snedden is from UCSF, and she studies the underlying biology of the pancreas in the development of type 1. She and her team are working to develop a deeper understanding of the identity and biology of non-epithelial niche cells within the pancreas. And they're doing this to enable a more directed and efficient way to replace lost cell and organ function via regenerative medicine. And Dr. Snedden also has a deeply personal connection to type 1. She has a very good friend from college and also from high school who were diagnosed with type 1. She is also the recipient of the very prestigious Helmholtz Young Investigator Award. So with that, I would like to invite Dr. Meyer to speak first.
Thank you, Karen. I'm going to share my screen and PowerPoint presentation. Can everyone see this okay? Excellent. Well, thank you for the opportunity to speak today. I'm going to cover kind of a general talk with some specific limited data, not overwhelming, hopefully, about cellular immunotherapy as a strategy for inducing immune tolerance through two basic pathways. One is by using bone marrow transplantation, the idea of having a coexisting donor and recipient immune system. And the other is modifying regulatory T cells, which you've probably heard before as something that UCSF has really pioneered in the type 1 diabetes realm. So many of you have seen this slide, which generally depicts the different stages of progression to type 1 diabetes and then of beta cell decline. I think that one of the key points, and this is something that my son has asked intermittently, and any time I've talked to the JDRF community comes up, is, what about the cure? How are we getting to the cure? And I think a point that Karen made is that there really are cures out there in the sense that there are methods for inducing long-term disease control without requiring supplemental insulin that have been discovered and are being moved towards FDA approval. In particular, the work of Jeff Bluestone and others goes back decades, has shown the strategy of depleting T cells, particularly as the disease is developing and for at-risk individuals, can divert the disease for some individuals for a very long time and potentially indefinitely, but not for everyone. Likewise, centers like UCSF, Edmonton, Miami, City of Hope have really, among others, pioneered the idea and concept, as well as clinical translation of islet transplantation, which, as Karen mentioned, there are individuals who are on immunosuppression but have complete control without exogenous insulin and are actually doing well. Both of these strategies will hopefully be approved and available to select patients. And over time, we'll see this strategy also grow with the advent, testing, and then commercialization of different approaches. And I think one of the things that's so exciting about the field in general, and also about the Center of Excellence, is this idea that right now we heavily depend on immunosuppression for these strategies. But really the preclinical data has shown now for some time in increasing complexity that we have new ways of doing immune reprogramming, that we can move from immunosuppression, maybe we use it for initial disease control and initial transplantation, but then it becomes more about using more sophisticated and deeper science, potentially cellular immunotherapy to reprogram the immune system so that you can have and stop responses without relying on immunosuppression. And that is, I think, what most people would describe as a goal of of a durable, long-acting cure. So the immune challenge of islet transplantation, as Karen mentioned, I started out really and I'm still in bone marrow transplantation, which the major indication is cancer. And we use replacement of people's immune systems by bone marrow transplant to replace cancer cells, but also to use the new immune system to fight and get rid of cancers. And that process is very difficult. In the case of islet replacement and type 1 diabetes, any new islet graft has kind of two problems. One, it has to get around the initial autoimmune attack that's highly honed and sensitized to the islet. And the second is that it has to also get around the alloimmune rejection that can occur from different constituents of the immune system. These are not necessarily the same actors. And this problem is as difficult as leukemias, if not more, in some extent, that we deal with in cancer. It's a very difficult immunological problem in question, probably one of the most difficult in medicine, as you may have heard before. But there's a lot of strategies to intervene. There are strategies to potentially limit these cells by using other cells. There are strategies to, as you'll hear from Julie, to engineer new islets that may have different features that protect them from the immune system. And also efforts to shape the alloimmune response, which you can shape because when you put islets in, that's when it starts. So I think it's a very exciting time if you think about the preclinical data again. So two promising cellular immunotherapies that are being applied to type 1 diabetes and islet transplantation actually actively now, again, in large part to UCSF, Jeff Bluestone, Kwasi Tong, and others have really pioneered the field, is the use of T regulatory cells, which are an immune constituent, like the police of the immune system, that help control and shape immune responses. And they've been shown to be very safe in the clinical setting in type 1 diabetes. And in the preclinical setting, especially with genetic modification of the future, potentially very effective in controlling and regulating the disease. Again, as we develop these technologies, it may not be true that every individual will achieve the perfect cure, but there are new trials that will come from this. And my lab in part studies this mostly in the context of bone marrow transplantation. Bone marrow transplantation may seem like an odd thing to talk about for type 1 diabetes, because in this context, you're taking a donor's immune system and you're putting into a recipient. And that process can be challenging. It can be subject to complications. For example, if too many of the donor cells go in too quickly, it can cause the donor cells to attack the recipient, and that can cause a major complication. There are also issues with infection, and there's also issues with conditioning the recipient to get the bone marrow in. So in general, it's been thought of as a method for fighting cancer and a nuclear option with very high dose chemotherapy being used. But one part of bone marrow transplant that's been discovered since the 1940s is that in animal models, and now we've shown in humans, if you can get the donor bone marrow and immune system into the recipient at a level where it coexists, you can then protect the recipient from rejection of islets, for example, and actually you can reverse autoimmune manifestations. So this is one way that we can develop immunological tolerance for islet transplantation. So going back 20 years, there were proof of concepts in mice that this could be applied to type 1 diabetes, looking at mice that develop insulitis in a model of type 1 diabetes. And Megan Sykes and Judy Shizuru at Stanford showed that you could do this kind of bone marrow transplant in mice, and if you could get sustained donor and recipient blood systems, you could reverse the autoimmune process and you could actually tolerize to a new graft, and the mice would not need immunosuppression. That's the home run. The problem, of course, is that the conditioning that occurs has traditionally been exceedingly toxic, involving radiation and high-dose chemotherapy. This This mouse model, which I show here, are experiments that we're doing more recently, in which on day 0, we do conditioning that's clinically portable and used in a clinical program for solid organ transplant. And this is a good mouse model for that. And then we follow it, we do the islet transplantation, and then we follow it with the bone marrow transplantation using this conditioning that we can test in clinical trials presently. And we show that the islets are retained for greater than 300 days and stable. And this, we use what are known as luciferase positive islets, which is pretty cool. They actually glow. So you can measure them and look in a machine at their persistence. And here you can see that mice that got islets but didn't get bone marrow transplant, the islets are rejected. But those that got islets and then did get a bone marrow transplant, you see retention of the islet graft. And when you add T regulatory cells to the mix, it can potentially improve that. And here we show a graph where we have the blood chimerism in the recipient after transplant. And this is at 300 days. And you can see that there is a mixture of donor and recipient, as shown here, in the different groups. This is a bone marrow plus islet, and this is the bone marrow plus islet and Tregs that lasts, it's approaching 1 year. So one of the major evolutions in the field is that conditioning for bone marrow transplant is getting much safer. So it used to be reserved for people less than 30, and now we actually transplant individuals up until age 80 even. So we're getting much better at getting that immune system in. And one of the major trends is also what Judy Shaziro and others are testing now is using, instead of radiation or chemotherapy, biological agents or potentially cell therapy agents to do that work without toxicity. That's a major frontier for our field. Judy's actually testing that concept in clinical trials. So this idea that I talked about that won the Nobel Prize for investigators going back into the 1940s and 50s is something that's been translated to the clinic, and I think very successfully as a pioneering center at Stanford. And in humans, what this means is individuals are conditioned. In our case, we're still using some radiation and a T-cell depleting agent or immune depleting agent. And the donor cells, bone marrow cells, are introduced. Over time, the donor and recipient cells coexist. And during that process, you get reprogramming of the immune system that can, again, potentially reverse autoimmune manifestations. And you get the induction and use of T regulatory cells to help control the immune system as well. So the idea here is that you can get immune tolerance with minimal or no immunosuppression. And as a proof of concept at Stanford and other centers, but really Stanford with the least toxic conditioning regimen, we've been able to do living donor transplant using the bone marrow from donors and the kidney from donors transplanted into recipients. And we've shown that we can get immune tolerance in individuals and they're off immunosuppression for years and doing quite well. That process depends on the genetic mismatch. So one of the big barriers is moving from individuals which were related and have the same immune genes to moving to individuals who don't have the same immune genes because the immune response is stronger. And we're in that phase right now and making steady progress. Another problem is moving from living donors to deceased donors, but we now presently have a FDA-approved clinical trial mechanism to use deceased donor bone marrow, which means that it could be applicable in the near future to islets and islet transplantation. So this is a very powerful immune reprogramming. The main limitation has been the conditioning regimen has been too toxic, but there are methods to do this conditioning potentially even on a non-toxic basis through innovation. So what we're doing in our lab and as a program is we're testing genetically modified T regulatory cells, and this is something that is being commercialized by companies and is very promising. We're also testing safer bone marrow transplant conditioning strategies, and my lab is actually integrating T regulatory cells into bone marrow transplant conditioning and islet transplantation is a way of synergistically using multiple cutting edge techniques to potentially induce immune tolerance. And just to show you some of the power in the field, I wanted to just give you clinical trial results. This is in the pure bone marrow transplant setting, but it shows you how putting these ingredients together can make things much more safe for our patients. In this clinical trial, these patients get very intensive conditioning, and many of them get a complication when their immune system, the donor immune system, takes over, again called graft versus host disease, that can be lethal. But when you actually introduce T regulatory cells into this transplantation, at our center at Stanford, we see clinical trial results where our patients have no clinically significant graft versus host disease, as compared to what one would expect at about 15%. And as a consequence, because the immune system is reconstituting better, traditionally, unfortunately, our patients die because of a complication of transplant at a rate of 11% per year. And trial participants at Stanford on this trial, and there are now 48 of them, we've seen no transplant-related mortality. This is actually true for a multi-center trial of now 70 patients around the country, which is pretty remarkable. And it shows that although this is not type 1 diabetes, we're developing approaches and techniques in other settings that can be pulled into type 1 diabetes to make what was inconceivable before possible and safe. And that's I think what kind of keeps us excited. So one of the things that the Center of Excellence has really done, aside from allowing me to interact with some of my scientific heroes, like Quincy Tong and Jeff Bluestone, who are really wonderful scientists in the field, interact with them more, is it's actually caused our teams to become closer. And one thing that we've done is on the clinical side, we've looked at combining forces between our experience with kidney tolerance and UCSF's outstanding experience as a leader in islet transplantation to say, not only could we develop these preclinical models, but could we translate them into a pilot clinical trial, which we've termed the UC Spirit idea, where we can test these concepts, not necessarily in 10 years, but maybe in 3 to 4, 5 years. And that's really exciting. We actually applied together for NIH funding, a very large grant that we're waiting to hear back from. And that's, I think, really honestly only possible in part because of the Center of Excellence and how it brought our teams together. And you can see here that in our grant application, we put the fact that we were integrated in a Center of Excellence as a key factor for why we thought this would be successful. And it's been such a pleasure to work with colleagues like Julia and others. Of course, one of the major problems in the search for the cure is that islet transplantation will only always be pertinent to a small population because of the supply of organs. There's a limited number of organs, islets, and bone marrow that can be used. But through the potential power of regenerative medicine, in thinking about how to integrate these things together, that it may be possible, who knows when, but that we can actually do this for many, many patients from genetically modified cell banks, which would be absolutely fantastic. And that would be the goal to shoot for. So I just want to, these are acknowledgements for just the data that I've shown. There's so many people that have been wonderful to us. And again, the support really comes from the JDRF, who's allowed me to do my career transition, which I always say, but when I went to my chief in bone marrow transplant and I said, I want to switch from cancer to type 1 diabetes, he kind of looked at me and said, really? Because there is no reimbursable procedure for type 1 diabetes. That's a risky proposition. And nonetheless, I've gotten a lot of support, particularly from the integrated approach that we're pursuing. So thank you for your time. I'll stop sharing and let Julie take over.
Okay, thank you so much, Everett. I always love listening to you talk, and of course love working with you as well. I'm going to try my best here to share my slides. Let's see. Okay, can everyone see my slides now? Okay, wonderful. Well, thank you so much for the invitation to be here with all of you tonight. It's really an honor to tell you about some of the work that we're doing in this center and communicate how excited
Julie,
we are.
we can't see your slides.
Oh, I'm sorry. Let's try it again. Can you still not see? Let's try one other option here.
If you want, Julie, I can also pull up your slides.
That's okay. Let's see if we can get it down.
Oh, there we go.
Is that better?
Yep.
Great. Sorry about that. I just wanted to reiterate how excited I am to be here to tell you all about the Center of Excellence, some of the work that we're doing, and really just a few highlights of what we're most excited about, you know, and want to just share our enthusiasm with you. So after Everett's beautiful explanation about the immune sort of tolerance side, I'd like to tell you a little bit today about what we're doing together as a center on the cell aspect and the engraftment aspect for a cell therapy-based cure for T1D. So I'd like to start off with this slide, which we had a really nice symposium last week at Stanford, which I was invited to talk at, and it was really nice to see the first speaker also invoke this quote from Frederick Banting, who was, of course, one of the discoverers of insulin in 1921. So it's the 100th anniversary now of this discovery. And in his Nobel acceptance lecture, which happened a few years later, he said, "Insulin is not a cure for diabetes, it is a treatment." And I think that really motivates so much of what we're doing here in the center and so much of why we're all here tonight, which is that, of course, the discovery of insulin was life-changing and life-saving, and it really transformed the field. But we are looking to do the next transformation because, as this flame of hope on the left from Ontario, Canada demonstrates, we really want to find a cure or cures for this disease, as Michelle, I think, so eloquently put. So what are the cell therapy-based cures that we're really excited about and moving towards in the center? Well, as I think many of you know, the real holy grail of stem cell biology has been cell replacement therapy to cure diseases. And in the case of type 1 diabetes, we know which cell type is destroyed. It's the beta cell, of course. And the overall goal here is that we could find a highly replenishable source of cells. In this case, we use stem cells to generate beta cells that could then be used for replacement of the missing or lost cells in the body. So we want to take diabetic patients who demonstrate hyperglycemia, and then through beta cell replacement therapy or islet replacement therapy, we want to return them to euglycemia. Now, this looks really sort of easy to put it on a diagram like this, but really decades of work, of course, has gone into this from many different groups, including ours and my colleagues, Dr. Matthias Hebrock and others. And it really was a culmination of incredible sort of tour de force of work. But what I'd like to say is that islet transplantation, as Everett said so nicely, really highlights the potential and shows us the light. It sort of shines the light forward towards this as an approach, because Mother Nature, we think, really does it best, meaning the ability of Mother Nature's made islets to control glycemia is incredible. It's this exquisite control. And you can see this very nicely in this set of plots. So on the left is a patient, I believe it was a child before islet transplantation, and each colored line is just a different day that they followed this child's glucose levels. And on the right, you can see after transplantation, the incredible amount of regulation that is achieved just through this procedure. So as I said, this shines the light for the forward path towards this type of approach. But of course, as Everett also mentioned really nicely, there are two key challenges that we have to address so that we can make this type of approach accessible to the millions of people who could benefit from this kind of therapy. So the first key challenge is that there really are not enough islets to transplant. And the second is the need for chronic immunosuppression. So in the center, we're working towards trying to clear these hurdles. And I'd like to tell you a little bit about, mostly the first one, because Everett just told you about some of these amazing work that he and others are doing to attack the second. So how would we clear these hurdles towards insulin independence? Well, we've identified 3 key hurdles that we think are really important as we think about progress forward. The first is identifying a highly replenishable cell source that can generate highly functional, I'm going to argue islets are the way forward, not just beta cells, but islets that can be used for this replacement. The second is, once we've made the right cell, we need to get it into the person, and we need those engrafted islets to survive. They need to be hooked up to blood supply, and they need to get the nutrients they need, and they also need to be able to sense glucose and function properly. And then that third hurdle, of course, is what you heard so beautifully from Everett just now, which is we need to make sure that these cells, even if we make the right kind and we get them into the body, that they don't get attacked by the immune system long term. So this goes back to what Karen articulated in the beginning, which are, I think, very ambitious and incredibly exciting goals of the JDRF Center of Excellence in Northern California. So here we are here in this picture. And the first is to just try to discover how immune cells and beta cells are interacting within islets and during the progression, initiation and progression of type 1. The second is to create islets and immune cells from stem cells as the basis for what we're thinking about as this next generation therapy or therapies. And the third is to develop an islet transplantation protocol, meaning stem cell derived islet transplantation protocol, ideally, that will induce tolerance and not require immunosuppression. So again, just have time for some very brief vignettes that I'd like to highlight to you. But let's talk first about some of the ways that we're approaching this first goal. So what I want to tell you about today is that it's an incredibly exciting time, both as a scientist studying these problems and also as someone who has a loved one with this disease, which is that we have this explosion of incredibly exciting, rapidly emerging technologies that we have really jumped on to leverage to study these problems, in particular to study islets and the immune attack, as I mentioned. So there's a lot of sort of nitty-gritty details about these approaches. So I thought I would use an analogy and we can try it out and see if it works. I've heard a version of this analogy before, but what I'll say is that this is a smoothie, of course, on the left-hand side. And if you're like me, you make smoothies for your kids, at least once a day. But really the limit of resolution essentially for what we used to have to understand the composition of a mixture. So let's say we want to study islets, would be essentially to grind those islets up, just like as you would grind up fruit for a smoothie, and then sample that smoothie, suck a little bit out of the straw. And then you might be able to detect things like a hint of mango, a bit of strawberry, a little bit of raspberry, but you would not know how many raspberries went in there, how many blueberries. You might not even know all the fruits that went in. And actually, in this case, understanding all the fruits and their relative numbers and their properties is essential for us to solve these scientific questions. So this former approach, which is not even that old, just a few years old really, but this is such a rapidly advancing field, was called bulk RNA sequencing, bulk, in aggregate. Well, I'm really excited that in the last few years, we were one of the first groups to study use a newer set of technologies, which I think is the equivalent of getting this fruit salad platter, where now you actually at least know how many fruits were involved, which ones. You still don't know all the properties of each of these fruits, but this is a really amazing transformation and incredible level of resolution beyond the previous one. So this is called single cell RNA sequencing. You could think of it as single fruit sampling. And so this is what I'd like to tell you about some of our incredible, advances that are going on now and some of the data that are generating and why we're excited about it. I also want to highlight what I think is the sort of present and future, which is now this fruit chart, where now layered onto this previous single cell RNA sequencing, we have something called spatial transcriptomics, which lets us know not just which fruits were involved, but also where they were in the tissue. So, or in the tart in this case. And that spatial information is critical because you may know, for example, that there's a lot of debate and uncertainty about whether and why some islets are attacked first or at all by the immune system and what is mediating those interactions. So we really need to understand that. So here's some key questions we're using these technologies to address in the center. One is, what are the critical cell types, the fruits, the berries in the human islet, and how do they interact with one another? That's really key. And secondly, why and how are islet cells attacked by the immune system? So using this fruit platter, that version, because we're still working out the technical details of the fruit chart, that's a pretty tricky one. I want to tell you about this first cellular atlas that we have generated of developing human pancreas. And you might think, well, gosh, isn't this known already? And we sort of thought that too in the beginning, but it turns out, no, that has not been discovered. And so we're really excited to contribute this to the scientific community. So what are we looking at? On the left-hand side, we have a plot. It looks really complicated, but actually it's in some ways very simple. So every single little dot on this plot represents a single cell, a single fruit, if you will. And we've now grouped these cells through these algorithms that look at similarity of cells to one another. We've now grouped them together and color-coded them, and we understand now through a lot of different scientific research and annotation that they belong to different groups. And you can see these purple cells here are the immune cells in the developing human pancreas. We have the endocrine cells, so this includes the beta cells, the alpha cells, etc. And we know what cells they are because they express certain key genes that gives us these clues. So this is the first time we have a cellular atlas of how Mother Nature makes islet cells in the body, in humans. And that's something that we didn't know before. And we're finding that although the mouse has been an amazing model for us, in the field up until this point, there are key differences in how Mother Nature makes a human beta cell versus how Mother Nature makes a mouse beta cell. So we're really excited about this data set. I could represent these data in a different way, which is simply just to show you a colored box for every new cell type. And you can see this incredible array of different cell types. And so many of these are not known. This is 144 cell types that we've identified so far. So this is, I think, very exciting. And one of the ways that we're using this data set, just to give you a sense of that, is now we're comparing Mother Nature's method to the method that we use in the dish to make beta cells from stem cells. So I'm showing that here where we took in vitro, just in the dish, cells we sampled throughout the directed differentiation we call of stem cells. So that's basically in the lab are the blue cells at the top. And then we took Mother Nature cells, the red, and we merged them together and we can figure out, for example, which genes are getting turned on incorrectly or turned off incorrectly, and which genes look like they're on track during this process of generating a beta cell, this differentiation process. And it doesn't matter, the details about the genes, but just to give you some examples of some of these genes are highly concordant over developmental time and some are really different. And now we know which ones to focus our energies on to sort of correct and make fine tunings in order to improve what we're making at the end. So those are some ways, just brief highlights as to how we're trying to make these discoveries about how these cells interact. What about our methods and our approaches for generating islets and immune cells from stem cells? I did want to touch on one set of approaches that we're using that we're really excited about. So this is to highlight the following fact. In the body, beta cells do not exist in isolation. We've been making them for many years in the lab as sort of balls of beta cells that come from stem cells, and that's a really incredible advance in and of itself. But really, I think we need to call our attention to the fact that in the body, these beta cells live with other endocrine cells. They live with alpha cells. They live with cells that line our blood vessels, the endothelial cells, and they live with other cells that constitute what we think of as the tissue microenvironment, in other words, the niche. And so one of the things we're really excited about is building this islet, not just a ball of beta cells, but an islet, because really the islet is the functional unit that regulates glucose homeostasis in the human being. So we want to generate an islet, but how would we do that? We really don't have the blueprint, right? So we're excited to understand first, what is the blueprint that Mother Nature uses? Well, we don't know all the details yet, but we do know something really exciting now that we didn't know about a year ago, which is we now have very high resolution, 3-dimensional imaging of human pancreatic islets. And I can show this to you on the left. This is a mouse islet. And this is a collaboration with a wonderful bioengineer at UCSF. And this is a human islet on the right. I think you can already appreciate, without going into too many of the details, that there are a lot of differences in the shapes of these cells, their relationship to one another. And so we're very excited to quantify this and use this as part of our blueprint. And another way of showing you these data is to then take these projections and rotate them in 3 dimensions. And hopefully what you can see is that we have the aggregate islet on the left-hand side, and on the right-hand side, we can basically make molds or casts, essentially, to represent the vasculature, shown here. You can see this incredible intercalation of the blood vessels. That's really important to the function. some of the beta cells now highlighted here on the right. And also in a minute, you'll see some of the alpha cells. So this is an incredibly exciting set of approaches, I think, to generate for the first time a high resolution 3D blueprint so that we can actually go from the left to the right. All right, but now that we are generating the blueprint, how would we actually build that islet? That's still a challenge. So we are using tissue engineering strategies to do so. So we generate our beta-like cells, BLCs, from stem cells, as you can see here. And then these cells, we just bust them open, we break them apart. We let them form together with a bunch of other cell types that I told you about that constitute that niche or microenvironment. And these form what we think of as engineered islets now. It's all human cells that we're using. This is what they look like after a few days in the dish. You can see insulin is glowing green here. And what we see is that when we include the niche and we make these engineered islets, they show improvement both in the dish, which is important from the perspective of function, but also when they engraft, when they function, and also when they rescue diabetes in rodent models. So this is just one data slide to show you this finding, where you can see if we have beta-like cells alone on the left-hand side, after just 7 days, we've lost so much of that input graft. Everett did a really good job of talking about the cells glowing. So we're measuring the amount of glowing, which is proportional to the input, proportional to the number of cells that are left. And in the middle, we have our beta-like cells with niche, our engineered cells, our engineered islets, I should say. And you can see really retention of nearly all of that initial graft. And similarly, when we look after 6 months, it's a long time to follow a mouse, but after 6 months, we can see quite a bit more human insulin still being made by the engineered islets that have the niche. So that's just one very, very quick highlight of how we're generating islets. I didn't touch on the immune cells, but we're happy to talk about that in the question and answer period. And then you heard beautifully, I think, from Everett about some of these extremely exciting approaches to induce tolerance that don't require immunosuppression. So I'll just leave you with some ways that we're really trying to investigate cell source. And now we have a benchmark for understanding on a single cell level how close we are. I think the answers were really close, but we know a few things that we want to really fine tune to generate beta cells from stem cells. In terms of graft survival, we and others have identified ways to really get those engrafted cells to survive and function long term. And then immune rejection, you heard from Everett, I stole his beautiful little Treg picture, his T regulatory cell, the mixed chimerism approach. We didn't talk about this, but in the center, Drs. Hebrock and Perrin and Tong are also using CRISPR-Cas9 genome surgery to make these cells cloaked so they could be protected from the immune system potentially. And then also there's the possibility of encapsulation as another approach as well. So the last slide is just to leave you with the flame of hope. This is something that burns brightly inside all of us, and I hope all of you as well. I'm extremely excited about the approaches that the center is taking, but also this opportunity that we've had now to synergize in ways that just weren't possible before the Center of Excellence brought us together in this really exciting way. So this is my acknowledgements. Thank you so much for your attention, and I think Everett and I are happy to take some questions as well.
Thank you, Julie and Everett. Those were two amazing presentations, and they really highlight not only the 3 projects that the center is working on, but also how your work really ties together so nicely. So I appreciate you taking the time to do that. And I also just wanted to express appreciation to both Matthias Hebrock, who is one of the co-leaders of the Center of Excellence, and Jeff Bluestone, who's also involved in the Center of Excellence. They've been putting little shout outs in the chat. So I appreciate both of you, Matthias and Jeff, spending your evening with us. And I'd like to encourage anybody, if you have questions, to put it in the chat, and we will make sure to get those questions to Everett and to Julie. But while you're doing that, let me just ask you, the question that always comes up, and oh, by the way, we did get a ton of questions as people registered, but I think one of the more common ones was just, what are you most excited about, kind of both short-term and long-term? What do you see happening in the center and in your work short-term, and then what excites you for the vision and the potential long-term?
So I'll start, and Julie, if that's okay. I think that what really is exciting is a combination of the preclinical studies that we're doing, where there's just amazing like conceptual framework in mice about how to protect, how to engineer cells, how to do these procedures in a way that you don't need to use immunosuppression. And then also thinking about, well, in 3 years, can we be in meaningful trials to test these things in select populations in a safe way? It's almost like the probes to Mars, right? Building that and launching it and making progress concretely by focused efforts where each of the teams are thinking then about the next step is so exciting. I'm very excited about Julie's work and Kyle's and Mateus's. The nerd in me just goes crazy every time I see what they're doing, because that's the future. I mean, I like the idea of getting, for me, in my career choice, getting to the clinic and clinical trial is what I've chosen to do. And that means very proximal efforts. But when it comes to the long-term, as we like to say, off-the-shelf solution for many, many patients, it's what Julie's doing that's so exciting.
I would just echo everything Everett said, as usual. I 100% agree. I would say that at a high level, I just want to also highlight, I'm so just genuinely excited about the power that comes in the union of and the synergy of these very basic biological approach, very basic research, combined with this incredible clinical expertise and the ability to actually get things into patients and into clinical trials in ways that are effective but also safe. And there's an incredible amount of savvy and knowledge that needs to happen on both sides. And I feel extremely fortunate to be a scientist studying in this area, but to have these colleagues who are just unbelievable at all of these different aspects. And you need both aspects, I think, to propel this forward in the way that we want to see, right? It needs to be guided by the basic science and the sort of continual innovation and iteration that has to happen over and over and over again in the lab. And you absolutely need the expertise of people who actually understand the considerations of getting it into patients. So I'm really excited about that synergy. And, it's sort of like, I don't really watch much TV myself, but I know some people like to watch cartoons where, there's sort of the superpowers that people have, and one person is fire and one is ice and the other is wind or whatever it is. And then they come together and they build a sum that's greater than the whole of the parts. And that's really what I feel like we're doing in the center with the potential to continue to do, which is just leverage and synergize and create value in a way that's not otherwise possible with siloed research programs or even siloed universities at this point.
Yeah, actually building on your last comment, Julie, do you both have a couple examples of how the center has enabled collaboration that's actually led to some of that kind of the sum of the whole being much more, adding up to much more than the individual parts?
Yeah, I mean, I think the crux of it really is what we've been talking about tonight already, which is that we need the cell source and we need the induction of tolerance. We want the induction of tolerance as well, right? To free people from the need for chronic immunosuppression, and that those types of expertise are actually not typically found in any one lab, right? Or even necessarily in any one department. And so that's the crux of it. I would say there are some really exciting sort of interactions and collaborations we've had cross center as well that I'm really excited about. For just example, there's another researcher who's part of the center who's at Stanford, Kyle Lowe, who has been doing incredible work on derivation of all sorts of specialized cell types from stem cells. And before COVID anyway, I used to hop down to Stanford and meet with Kyle and his group. And we have, we're really excited about some plans to combine, for example, those niche cells that I was talking about, combine his version, which are made from stem cells, in order to do sort of a super version of what I just described, where we could get a single cell source. Could even be patient-specific IPS if we felt there was value there, but it would be a single cell source that we can be using to generate the beta cells and the different niche cells. So that's one example that was incredibly, just stimulated by the Center of Excellence interactions. That's just one small example. Everett, I don't know if you also want to chime in on other specific examples that you've experienced.
Yeah, no, I think the lines of communication, just seeing what people are doing opens your eyes and allows you to contextualize. But also working with colleagues who are so experienced, when you run into an issue or problem and they're just, you know, a Zoom call away and there's a way to interact, it just allows us to progress more quickly. And I've relied on colleagues at UCSF and at Stanford for those little things which really add up. Also, you know, to a larger perspective, I'm so excited to be able to interact with Greg Zott, Andy Passault, Quincy Tong. These people have been in the islet transplantation field and the T regulatory cell field and are experts par excellence. And I'd like to say that we're experts in immune tolerance and combined bone marrow kidney transplant. But together, as Julie was saying, the elements mixed and combined and are stronger. And that, being a part of that, and I have to say, as humbly as I can, I think Stanford and UCSF are very unique in those skills in the field, and to have two centers very unique and excellent coming together. It's kind of like the Wonder Twins, you know, it's just, there's so much more you can do. And also on a personal level, you know, as I talked about, JDRF has really supported my career transition, which is always a work in progress. But, you know, having Sung Kim, for example, at Stanford, sitting down with him and saying, what's our vision? And then being able to align that with UCSF and have that camaraderie and friendship. And I will say it does extend beyond our institution. So when we talked about our efforts, we are including institutions around the country, because if we come up with something, we can test it, but you've got to test it elsewhere. And that's also something that's been very important as part of what we're doing.
Okay, I know we're coming up on time, but we have one more question from Dick Allen, who is also an incredible T1D champion. And he asks, I think for Julie, researchers at the Joslin are doing some work on modifying beta cells to, quote, remove the offending genes and replace them with good genes. Is this at all similar to the work that you're doing?
Yes, thank you for the question, and thank you for being a champion. We need more champions like you. So the answer is yes. This is actually a big part of the Center of Excellence, and we are doing some of this, but I also want to give a shout out to Drs. Hebrock and Parent and Tong, who are also using this approach. So that's what I meant, and I probably wasn't very clear, but that little image that I had of the genome, the DNA, and the scissors, that was meant to communicate that we're using this approach. People call it sometimes colloquially genome surgery, and the idea is at least twofold. So number one, we are using it in the center to try to induce tolerance to donor antigens and also to enhance graft survival. And I liken this, if you are like me and you've been reading Harry Potter to your young children, we just finished the series actually with my older child. I think about the invisibility cloak that Harry Potter has, which really gets him out of a lot of scrapes, right? So it's very similar, where we want to sort of change the DNA just a teeny tiny bit, such that the immune system either can't see the cells that are going in, or that the immune system is a little less offended by them, if that makes sense. So that's one way the invisibility cloak approach is something that you're talking about in your question. And a second aspect of the answer is that there's also an approach being worked on in the field, which is to insert what are called kill switches. And it sounds rather negative, but the idea is actually a positive thing, which is to rapidly eliminate cells post-transplant. So if there's any concern about overgrowth or tumorigenesis or anything like that, or you just want to get rid of them for some other reason, there's a drug-inducible kill switch option that you can encode using this genome surgery again, that could potentially let you do that. And there's actually companies working on this approach as well. And there has been proof of principle published. So those are two ways that, yes, we are thinking about and doing those types of approaches. And it's a very good question. Thank you.
Well, thank you both. You're getting a huge number of shout outs in the chat. Everybody's saying thank you and really excited about the work that you're doing. And I also wanted to thank Dr. Sung Kim, who's the co-leader of the Center of Excellence from Stanford. He's also attending. So with that, I think we'll turn it back to Michelle to close up the session.
Thank you. I offer our collective thanks to you, our speakers. It's an incredible opportunity for all of us to learn from 2 of our 13 exceptional scientists at the center. And thank you too to our attendees. We're here today because of what you've done to support JDRF. And thank you especially to those of you on the call who are already center supporters. So we need your support now more than ever, whether it's being a volunteer, whether it's advocating for JDRF's important initiatives, whether it's participating in a clinical trial. And of course, I couldn't help but see some big WALK fundraising team representatives on our call. If you're fundraising and donating to JDRF, JDRF has made a commitment of $8 million over 5 years to the center. We've raised $5.9 million, and we hope to home in on our goal of $8 million in a short amount of time. So hopefully you share our family's excitement based on what you heard today, and will be interested in helping us support the center. We will be accepting directed donations of $10,000 or more, which is unique for JDRF, but this is, again, such a unique opportunity that we have. This couldn't have even happened 5 years ago. So we're very lucky to be in the position that we're at. So JDRF staff are available to answer any questions that you might have. And we just want to say again, thank you to all our supporters. We are where we are because of you. And I'm going to take Julie's flame of hope, last slide, and say, let's keep the flame of hope alive. Thank you.
Thank you, everyone.
Thank you so
Right.
much, everyone.